Hi everybody, and welcome back for another instalment of Teach Me in 10, the video series that is brought to you by LabTube, which is part of the Technology Networks group. My name is Molly and I'm a senior science writer here at Technology Networks. Our Teach Me in 10 video series invites scientists to be our guest and talk us through a scientific research area or a scientific concept in less than 10 minutes. The aim is to make science as accessible as possible for you. This week, I'm joined by Dr. Yager from the University of Albany. Dr. Yager is going to be talking us through the polymerase chain reaction, also known as PCR. How does it work, and why might it be used?
Thank you, Molly, for that kind introduction. The technique that I'll be explaining today is pretty powerful. It can help solve crimes. It can help diagnose and treat disease. It can help in identifying microorganisms, both in the environment and ones in our bodies that might cause disease. It can also help us unlock the secrets to our own genome. What is that technique? That technique is polymerase chain reaction, or PCR. This is a method used pretty routinely in research and in the clinic to amplify DNA. We can take one copy of DNA and we can amplify that into millions to billions of copies. Now, why would we want to do that? We want to do that because by having more DNA, we can visualize it, we can manipulate it, and we can understand it. So PCR, as you'll learn, is a pretty powerful technology. Now, how is PCR performed? Now, there's a certain recipe for this process. We need a few things. First, we need a source of DNA. We call that our template. And that can come from cells or tissues. It can come from the environment. It can come from a crime scene. And really, it's our source of genetic material that we're going to assay. Second thing, we need our primers, and primers are very important. They give this assay its specificity. We're trying to amplify not the entire piece of DNA, but just a small portion of that, or a sequence or a gene. The next thing we need are dNTPs, and these are the building blocks. You can kind of think of those as Legos, if you will. We need to, if we're needing to amplify or make more copies of our DNA, we need building blocks to do that. And the last piece is a Taq polymerase. Taq polymerase is a specialized enzyme that is able to copy DNA. And it's very similar to the enzymes found in our own cells. As our cells copy DNA to make more copies of the cells, they use a similar enzyme. This enzyme is called Taq polymerase. Let's go through those steps in the PCR reaction. The first step is denaturation. You might recall that DNA is a double-stranded molecule, and this process can occur when the molecule is in a double-stranded form, so we need to break it into single strands. And by raising the temperature to 95 degrees Celsius, we can separate that double-stranded DNA molecule into two individual strands. The next step is the annealing process. So we lower the temperature from 95 degrees to somewhere between 55 and 65 degrees Celsius. And this is the appropriate temperature to allow our primers to bind. And in this diagram here, those are the red lines. And as I mentioned, the primers give us a specificity. It allows us to figure out which gene we're trying to amplify. Is it a gene important in a disease state, or if we're trying to understand physiology, or is it a gene that might be expressed by a bacteria or a virus if we're trying to diagnose a disease? So the primers at this temperature bind to our DNA strands. The next step is the elongation process. So our Taq polymerase comes into play. It binds to the complex of our primers and our single-stranded DNA molecules. The temperature is raised to 72 degrees, and this allows the Taq polymerase to do its job. As a polymerase, it creates or adds to or elongates those strands to create a duplicated copy of our DNA. Now, an important thing about this Taq polymerase is that it comes from a special organism. Now, as you'll learn, this process of PCR, we need to change the temperatures frequently, very high to very low. And if we used a polymerase that came from our own cells, that high temperature I first talked about, that step one, that would destroy the enzyme. So scientists needed to identify an enzyme that is able to handle those changes in temperatures. And this polymerase, Taq polymerase, comes from a bacterial species found in hot springs. So the way that that bacteria has evolved, it's able to handle those temperature changes quite well. So that's the steps in the process, and these steps are repeated over and over again for many cycles, 30 to 40 different cycles, so that when we had that first cycle, we were finished, we had one copy became two, two becomes four, four becomes eight, et cetera, et cetera. And if we do those 30 or 40 cycles, we get to our millions and billions of copies. Now, as I mentioned, this process requires temperature changes and cycles, so we can't run from a water bath to water bath to water bath. We need a better way to do this, a more efficient way. And as this image shown here, this is what's called a thermocycler. So in our events here, the recipe, we're adding in the DNA, the nucleotide, DNTPs, the primers, everything together in a very small tube. Those are put in this thermocycler, and the thermocycler controls those temperatures for us so we can go through those cycles in a period of time. And at the end, we're left with DNA, as I mentioned, that we can manipulate or visualize. For example, we can visualize an agarose gel as shown below. A particular example I'd like to share with using PCR is pretty relevant to our current COVID-19 pandemic, and that's the important role that PCR has played in diagnostics. So PCR is considered the gold standard for diagnosing COVID-19 or SARS-CoV-2 infection. Some of you listening to this video may have learned about this process or maybe have experienced it, but the first step is to collect the genetic material to sample from, and it's usually done by a nasal swab, throat swab, or saliva sample. And again, the purpose here is we're looking for the presence of the virus, more specifically the presence of viral genetic material. Now, the SARS-CoV-2 virus is an RNA virus, so there is an additional step that has to take place before PCR can truly be performed. We need to convert that RNA into DNA, so the steps that I just discussed can occur. Another difference here is that in terms of visualization, what's often done is that a special chemistry is used to add a fluorescent signal, so that if the DNA is present, if the viral gene is present and it's amplified, a fluorescent signal is detected. Now, this type of technology is performed in a specialized laboratory. It can take days compared to those at-home rapid antigen tests that some of you might be familiar with. But again, this test is very specific, very sensitive. And in terms of, you can detect a very small amount of virus, so we're able to detect the infection very early in the process, which can be important in making sure that we mitigate the effects of this pandemic. And because of those primers, this makes the assay very specific, so there are very few false negatives in this process. Now, this assay is not without its faults. For example, it can't distinguish live versus dead virus. So if you've cleared the infection, it's been weeks after you feel symptom-free, you're allowed to go about to school or work, you might still test PCR positive because the assay is so very sensitive, it could theoretically detect a single copy of the virus. So that's one of the things that need to take into account when we use this in a diagnostic sense. I hope you learned and gained an appreciation for the PCR technology, and I turn it back over to you, Molly.
I'd like to say a huge thank you to Dr. Eric Yager for joining us at Teach Me in 10 and for giving us a really wonderful overview of PCR. If you'd like to learn more about this technique, make sure that you check out the resources we've listed in the video description. We'll be back next week for another instalment of Teach Me in 10, but if you can't wait until then, make sure that you check out our LabTube channel where you can find all of the videos that we have published so far. See you next week.